Hello everyone, in this video, we will talk about plant in vitro cultures as biotechnological approaches for sustainability. In vitro culture, literally meaning in glass, refers to the growth of plant cells, tissues, or organs outside their natural environment, under sterile conditions and in specially prepared nutrient media inside test tubes, flasks, or petri dishes. Plants have a great ability to grow in vitro and develop outside their natural environment under controlled laboratory conditions. This is possible because plant cells are totipotent, meaning that every single cell within a plant contains the complete set of genetic information and the inherent capacity to regenerate and form an entirely new plant, provided that it is supplied with the appropriate balance of nutrients, growth hormones, and environmental conditions such as light and temperature. There are several types of in vitro cultures, each serving different purposes in plant biotechnology. Callus culture. It involves growing undifferentiated plant cell masses from explant tissues on a nutrient medium with specific hormones. These calluses can later be used for plant regeneration or for the production of valuable metabolites under controlled conditions. Organ culture. Specific organs like roots, shoots, or embryos are grown to study development. Cell suspension culture. Cells grown in liquid medium are useful for large-scale production of compounds. Micropropagation. It is a widely used plant tissue culture technique that allows the rapid multiplication of genetically uniform and disease-free plants under sterile and controlled laboratory conditions. This method ensures large-scale production of healthy plantlets in a relatively short time. In vitro cultures are not only useful in laboratories, they play a vital role in addressing global challenges such as biodiversity loss, climate change, and sustainable development. By propagating rare and endangered species in controlled environments, in vitro culture helps preserve biodiversity that is under threat from habitat destruction and climate change. Micropropagation enables rapid multiplication of trees and crops, supporting reforestation programs for carbon capture and ensuring resilient food systems under changing climate. Cell and tissue cultures can produce valuable bioactive compounds, reducing pressure on wild populations and providing sustainable sources for medicines, nutraceuticals, and green products. In vitro cultures offer the advantage of producing planting material that is free from diseases. They enable the rapid multiplication and propagation of crop varieties. This technique is valuable for developing plants that are well adapted to challenging environmental conditions, such as drought, high salinity, or heat stress. When natural disasters wipe out plant populations, tissue culture offers a fast way to restore species and ecosystems. In vitro systems allow scientists to study plant responses, introduce beneficial traits, and develop crops suited for a warming world. Plant in vitro cultures show that every plant cell holds the power to regenerate, and by harnessing this, we can conserve species, strengthen agriculture, and build a more sustainable future. Join us in this amazing journey. For more information, visit our project website and follow us on the Bioshield social media.